So this is where we're starting with, but we're going to go over a little bit of what we just did. So what's an allele? It's a version of a gene, right? So you can have many, each person has two alleles, one from their mom, one from their pop. It can be the same allele, it can be a different allele. There are oftentimes thousands of different alleles for one gene. So which of these can't belong to a child of this couple? Huh? Let's see here. Hold on. There's mom, there's dad. There's mom. There's. Oh. So it's C, right? Okay, it's C. So if you look at C, we've got mom up at the top. We've got mom up at the top for C. But we've also got mom, the lower gel thing, and we've got mom for that. Now, that could be an offspring if it was a clone of mom, right? But beyond that, it's not mom and dad. Because as you can see with dad, he's got the upper allele and the lower allele, and C doesn't have either one of those. It's not C. Okay. Okay. Go on. Which one for this one? Huh? So which cannot be a child of this couple? No, okay, so B, so we've got mom, and then we've got mom again, okay, and dad's not in there. In C, we've got mom and dad both have the same allele, that's a double, okay, and then D is the same thing. No, hold on. Yeah, okay. That's right, okay. So that's it.
How can D not be it?
Well, D doesn't have mom's allele.
Oh wait, we can because
B
it
can.
has
Dad
the top
and
one
mom
is
share the
from
same
dad
top
Oh, that's,
and the bottom from mom.
Right?
no, see, D has a double at right across from dad. And
No,
there's,
I think the B.
and there's,
B.
what?
B.
Oh, B is
B
right.
is a trick kid.
Yeah.
Yeah. So B is right, is the one that, but at the same time, D, it only has the allele for dad, and mom doesn't have the same allele there, so they're both wrong.
Just a quick question. So for B, even though it has an allele of the mom, it has the allele of the mom twice. Is that okay? Because
No. So what you have to do is, like on C, there's the same allele for mom and dad. They share the same allele.
So then B wouldn't be one for
But see that for the
Right.
B child, it has the allele, the top allele for mom and dad, and then it also has bottom allele for mom. So technically it has two alleles from the mom.
Oh, that's right.
So is that still a child that's still born?
No, that's actually good. That's actually still right.
It's still good, okay.
Yeah, so B is still right. Because if you notice, you get the top allele from dad, you get the bottom allele from mom.
Okay, so it doesn't matter that mom has the top allele too?
No. And then C, of course, is also right. So D is the only one that's wrong. That one's a little tricky. Okay, this is a review. Eukaryotic cell, a prokaryotic cell is either bacteria or archaea. So what kind of cell is this? Huh? It's a eukaryote. It's got a nucleus. Okay? Prokaryotes, in other words, bacteria or archaea, don't have a nucleus whatsoever. So this has everything. This has a nucleus, it has mitochondria, and then a bunch of other stuff that you really haven't learned about, but it's part of a eukaryotic cell. So who are these guys? Watson and Crick. Okay. What's the complementary sequence to this? What's the one for C? What's the one for G? G is C again. What's the one for T?
A.
What's the one for A? T. Okay, good. Okay. The double-stranded DNA molecule unwinds, as you can see. And then there's an enzyme that adds complementary base pairs. Do you remember what that is? Huh? DNA polymerase? So an investigator finds a small amount of tissue at a crime scene. What procedure will a forensic scientist perform to amplify that DNA? Starts with a P. Another enzyme. Polymerase chain reaction, PCR. Okay, so these are short tandem repeats again. Why are STRs so useful for forensics? What's one of the reasons? Who do we want to find out about? Anybody? What do you think we want to find out about? Come on. It says forensics there. Okay, so we're dealing with dead people, right? Or maybe live people. So who do we want to find out about? Think about crime. Think about natural disasters, right? We want to find out who the dead people are so we can match them up. We want to find out perhaps if the criminal has left behind some of his own DNA, right? Okay. Okay. And then this is a gel electrophoresis, remember? So we have these DNA fragments that we've, these STRs that we've duplicated with PCR. We put a different sample from in each well. If we apply electrical charge, which fragment will go down closest to the bottom? Huh? Well, it's always going to go to positive, but in terms of size. What? Larger takes, it doesn't go down as far. It's heavier. Okay, so a larger fragment is going to be up at the top. Okay, a smaller fragment is going to be, there's not very much of it, so it'll go farther down to the bottom. So you can see larger fragment. Then there's another one that's almost as large, and all the way down to the bottom where you have small fragments move quickly. Okay. Now, genes make proteins. This is what's called the central dogma. You can't go from a protein and translate it back into a gene. That's just the way it is. Now, that's not entirely true in terms of RNA, but for now, we're just going to say you can go from one to the other, but not back again. Okay? So the DNA is basically kind of an instruction manual. You transcribe that and then translate it into a protein. And the protein that you make, you can have a gene that makes a number of different related proteins. Okay. So essentially, a gene doesn't do anything. It's just like writing. You know, you write, and then if you know how to read, you know what it means. Okay. So how do genes encode proteins? Now remember, a gene, there has to be for each, now protein is made up of what? Huh? Amino acids, exactly. So these are all amino acids down here. There are 20 amino acids that we talked about. You don't have to remember every one of them. A codon is like, we'll say, A, T, T. Okay? And that's a codon. That's 3 different letters from the genetic code, right? In this case, it's ATT, and it codes for a certain amino acid. So a gene has a string of codons. That means 3 letters for each amino acid that is then transcribed and translated for each protein. And a protein, of course, is made up of amino acids. Okay. Now, what happens here is first we're actually making a copy of the DNA, and we're making it out of RNA. It's called messenger RNA. So this is mRNA. So let's say you have a DNA that's ATT. You then make a copy of it. You transcribe it. But in this case, RNA, you use the letter U for a T. It uses a slightly different peptide. Okay? And you make a copy of that. You then take this mRNA right here. You ferry it outside the nucleus. You put it into the cytoplasm. And then there's this thing called a ribosome. It's made out of different kinds of RNA. And it's going to translate that RNA into an actual protein. Okay? So in movement from the mRNA from the nucleus of the cytoplasm through a pore. That nuclear pore is some protein. And then it's going to run through the ribosome, and the ribosome is going to take these various amino acids and string them together in the right order to make the protein. So this is the way it kind of goes. In the nucleus, you have transcription. You go from DNA to RNA, messenger RNA. And then, in the ribosome takes various amino acids that are floating around and strings them together based on the mRNA. Mm-hmm.
So when it, after the translation, it makes the protein, it makes whatever protein it's supposed to do. So like for all different types of whatever needs to go on,
Yeah.
a bunch of different proteins that will be made.
Right. Yeah. So so the RNA is the template that's going to be used to actually put different amino acids in a certain order.
Okay.
And that corresponds exactly to what the DNA was.
Okay.
Okay? Okay, now, on a second. Okay, good. So, here we are, we have the DNA. We're transcribing it in the nucleus into this messenger RNA, and then we're translating it to the protein. So, transcribe is to write, type, or print, transfer information. Translate is to turn one language, let's say, into another. So we're going from the RNA, mRNA, we're translating it by stringing amino acids together into the protein that it's coding for. Okay, now, remember, what does each gene have in terms of a genome? Does it have a partial genome, or does it have a full genome? Does it have only the genes that it needs for itself? A full genome. That means every cell in our body has a full genome. All 22,000 genes. Okay? It's not like, an eye cell only has genes for making an eye. Okay? So, in terms of cellular differentiation, we use gene expression. Okay? Let's say, in this case on the left is a muscle cell. There will only be certain genes that are expressed out of the full genome for muscles. And then there will also be other genes that are expressed everywhere for like housekeeping genes. Yes?
So like you said, our eye only has certain cells or whatever to see, but it does have the full genome. But
So it has a full genome, but it only uses, in terms of creating an eye cell, it only uses a certain spot of that genome that only applies to eyes.
But it does
Have the full thing.
Okay.
Okay? On the right, we have nerve cells. This is the same sort of thing. So, in terms of cell differentiation, that means different kinds of cells in a multicellular organism. This is how it's actually expressed. So, only certain genes turn on to make a certain kind of cell versus others. But there are also other genes that are always used for, we talked about respiration, cellular respiration earlier, when we were talking about ATP. Remember what ATP is? That's the currency that the cell uses to do as energy to do work. Okay? So, those kinds of genes, cellular respiration, are used all the time, no matter what cells. The ones for nerve cells are only correspond to the nerve cells. Okay. So, in other words, all your cells have the full instructions. But different cell types use different parts of the manual. Okay? So, genes expressed in muscle cell. Look on top. Build. So you have to build different kinds of fast or long. Strong. Has to be the right kind of fiber. Heart is a muscle. And then you end up with a muscle. Now, in terms of, for the eyes, we still have build, but we also have dark. We don't have dark up on top. We have brown, and then we have eyes. Okay? So let me show this. There's a better way to look at it. Okay. So remember, each parent donates one chromosome. That means an egg is haploid. It only has one chromosome. One of 23 chromosomes from the mother. And it has all 23 chromosomes from the mother. The 23rd chromosome is the X, that's the sex chromosome. And the sperm is the same way. Okay? And then when you have a sperm and an egg together, you're back to diploid. That means you have a zygote. It has one copy from the mom, one from the papa. We have 23 chromosomes. 22 are homologous. That means identical. Doesn't mean the same allele, but they're identical in terms of what genes are on there and in the place they are. And then there's 2 sex chromosomes, an X for female, Y for male. Okay, so here we are, muscle cell. We have gene A. We transcribe it into RNA, and then we translate it into the amino acids and the protein that is made by those amino acids. Okay? Gene B. Also, not very much, you're not actually making very much of a protein, in this case protein B. You have one RNA and it makes 2 copies of, let's say, the protein B. Translates it. Okay, C. You have 3 RNAs and you transcribe into protein C a certain amount. Translate. So you see there's different amounts here. That's an important thing to understand because it's a matter of gene expression, how much each gene is actually expressed. And then we'll say this protein, no gene D is on. Okay? It's only used, let's say, for another protein. Okay, now let's take it here. We have a neuron. Now remember, some cells use the same protein, okay? Another cell and some cells only use certain proteins for the kind of cell they are. So let's take a look at
Professor, I have a question. So just making sure the amount of proteins that are given or shown represent the genes that are going to be shown or not. So if there's not, a protein is not given to a certain gene, it's not going to be shown, right?
it's not going to be expressed. They're not going to make the protein.
Okay.
Okay, so it goes from the gene up to the top and then translation, and in this case, the protein.
And no protein is given and that gene doesn't show.
Right.
Okay.
Yeah. Or the gene doesn't get expressed, and so you end up with no protein. It goes gene to the protein, so not the other way around.
Right.
Okay. Okay. In this case, we also have the gene is using, the protein is using, the neuron is using the A gene as well. Okay? So they share the same protein for some cellular function. Right? Now, in this case, compared to the muscle cell, gene B is turned off. There's no expression of the protein for gene B. Okay? None whatsoever. Okay? They both use gene C, but there's different amounts of the protein that are expressed. Okay? There's 13 on top of the individual proteins. There's 9 on the bottom. Okay? And then, in this case, gene D, unlike with the muscle, is actually transcribed and translated into the protein at the bottom. Okay? So this shows that there are some genes in this that are both being expressed in both types of cells, but not necessarily in the same amount. Gene A is translated into the same amount of DNA in both. But gene C is translated, there's more of gene C that is made in the muscle cell than in the neuron. And then in terms of gene B, only found in the muscle cell, none of that protein is found in the neuron, and then vice versa, gene D. Okay, now, they're going to, some of the reading you do, that they're going to say a gene is either turned on or turned off. It's not exactly like that. Remember, a gene is making a protein. It has to make the right amount of protein. Okay, it's not like, oh, I need this much protein, it's on, I don't need any. No, it's not like that. It needs, like we saw before, I think it was gene C, there was a different amount of the C protein that were made between the neuron and the muscle cell. So what it is it's more like a dimmer, a light dimmer. Right? Making it higher, making it lower. So it's not like on or off, it's like any, yeah.
But in the example you had just shown, for the muscle cell, gene D wasn't there at all.
Right.
And then the
Gene C.
gene D on the
On the other one
cell
was.
was there. So
Right.
I'm thinking,
Yeah, yeah. And a certain amount of that protein is expressed.
yeah.
Okay? So it's more like a dimmer, it's not like an on-off switch. So I turn the dimmer a little bit, it gets a little brighter, that means some protein. I turn the dimmer all the way to the end, it gets really bright, that means lots of protein. Okay? So this is how genes are expressed, and it's called gene regulation. This is why you can have the same genes coding for the same protein in different cells, but in terms of different amounts. Okay, so here's our lactase gene again. Now, it turns out that we want to know how does a gene actually get regulated in terms of how much it's supposed to express. In the simplest case, upstream from where the gene is, there's this region called the promoter region. Okay, this is what's actually controlling downstream how much of that protein is being expressed. Okay, so here we go. We've got this promoter region, and then we've got this coding region right down here. And this promoter region turns on this gene in terms of how much it is. And that, of course, that gene is called the lactase gene. It's an enzyme. It's to break down lactose into two different simple molecules of sugar, one glucose and one galactose. Or two sugars, a sucrose, which is a glucose and a glucose, and it breaks those down because what you need is you need glucose as a single sugar molecule for actually getting into the cell. Okay? So this here, the lactase gene, is what's used to actually do the transformation, breaking up a more complex sugar into a simpler sugar. Okay? And there's also what are called substrates where the actual reaction actually takes place. So this is a chemical reaction. And the substrates actually do have kind of a three-dimensional configuration that only allows certain molecules into it and not others. Okay. Now, the transcription occurs in the nucleus. Remember, we're going from DNA to RNA. The RNA polymerase binds to the promoter region. So you have this RNA polymerase, which is another enzyme. Okay. It binds to the promoter. and causes the gene expression down below. Okay, so here are the RNA nucleotides that are then being transcribed into mRNA. Remember, RNA is a messenger. It carries the transcription of the DNA all the way out. Yeah.
So this is what creates the mRNA, and then it leaves out and goes into the ribosome,
Right.
which then creates the...
But in terms of how much RNA is made, it's controlled by the promoter region. Okay? So, in some cases, the promoter region is like churning out tons and tons. There's no stop on it. There's other, now in terms of, let's say, lactase, there's a feedback loop. So if you have an infant that's drinking milk, it needs, of course, lactase to actually break that down into glucose. As long as it's getting fed lactose, it keeps that region turning on and making lactase, the enzyme for breaking down milk. Now, obviously, except for humans, all other animals, that's shut off. That promoter region is actually shut off when the animal is weaned. That means it doesn't need its mother's milk anymore. So, in terms of people who are lactose intolerant, they're just like young animals that are weaned and they don't make lactase anymore, and so they can't actually digest lactose. For people who are lactose tolerant, that is another form of food that they actually can digest and get important energy from. Yes?
So this happens before?
Yes. So in other words, as it's reading, as the DNA is being read, there's this upstream area that either expresses the gene that's coming later or expressing it in terms of how much is made.
So RNA and mRNA are the same thing?
So the mRNA, what it's doing is the promoter is actually, that's in the DNA.
That's when it moves out of the cell?
And no.
Got it?
No. So what's happening is the promoter is reading the DNA and it's telling how much of the mRNA for that gene is being transcribed. So it might be just a little bit, or it might be a whole lot. Remember when we showed that earlier slide about how much protein A was there and how much protein B? It's the same sort of thing. So the promoter, all it's doing is it's saying, oh, it's not going to make any of this gene, or it's going to make a certain amount of it. Okay?
Is RNA and messenger RNA the same thing?
Yeah, and there's a number of different RNAs. In this case, we're only talking about messenger RNA. In the cytoplasm with the ribosome, we're going to look at that. The ribosome is made up of a number of different kinds of RNA. Okay? And then, I'll tell you about it a little later, but it turns out that our genome actually makes a lot of other things besides this protein, which are, it makes regulatory RNA. In other words, RNA that will bind to some areas of the genome that will actually tamp down the production of that. But you don't have to know that. Yeah?
So red and blue make purple. So for the promoter,
Yeah.
it's kind of like saying it's giving 50% of,
Exactly.
and then the mRNA. What's the exactly the yellow stuff flying out?
Okay, those are the RNA nucleotides, like A or and T. You have U instead of T, or G, or C, like the same ones for DNA. Okay, and what it's doing is it's taking those nucleotides that are floating around in the actual nucleus and putting them in the right order for the RNA. Because those are RNA nucleotides. So remember, we have mRNA. The mRNA is made from those nucleotides. Okay?
I thought you said they were the same thing.
Those nucleotides are each individual base pair.
Right, the C, the G.
Yeah, exactly. And then the mRNA is made of all those different individual ones combined.
Oh, okay.
Okay? These are all good questions. I'm proud of you. No, because it means you're thinking, okay, this is how it goes. So it's good. Okay, now the central dogma is that you have a DNA, it's transcribed into mRNA in the nucleus, and then it's translated by stringing together these different amino acids in the ribosomes in the cytoplasm. You cannot go from turning a protein back into a DNA molecule. Okay? It's only a one-way street. Okay? This way, no way. Okay. Now, you might think, well, it would sure be nice if we could go back the other way, but this is probably arose because to prevent mistakes and prevent, let's say you could go from a protein back to the DNA, you could see how you could insert a mutation there by accident, right? So you don't want to do that. So that's why it's a one-way street. Okay, now, the translation happens in the ribosome. So this whole thing is the ribosome, right? And this is all in the cytoplasm. So remember the mRNA, this is the mRNA, right? That was the nuclear, that was coming from the nucleus out into the cytoplasm. And that's what's then being read to string together different amino acids. Okay? Now, it means that these amino acids are floating around. There has to be enough amino acids there for the ribosome to actually take and put in the right order. Okay? Now, in this case, there's another kind of RNA after the mRNA. It's called the tRNA, which is translation RNA. And it's actually taking each amino acid that it codes for, and it's translating from the mRNA into the appropriate amino acid, and then the amino acids are strung together in the right way to make that protein. Okay? So the translation occurs as it's a multi-protein complex. It's called the ribosome. Okay, and part of the ribosome is this RNA called transfer RNA or translation RNA.
So the cytoplasm is outside of the ribosome?
Yeah, exactly. So okay, we got a cell. We've got a nucleus, we've got the DNA, we've got the RNA that's made, right? The RNA is ferried out, and then in the cytoplasm there's all these ribosomes. Okay? So here's the transfer RNA again. This is the mRNA down here. This is the codon, remember? You have to have 3 letters. In this case, U substitutes for T in DNA. So it would be, for DNA, it would be TTT. This is UUU. It codes for a certain amino acid. In this case, phenylalanine. An RNA, instead of a DNA, we're using U instead of T. And the anticodon, in other words, the complement, is AAA. What?
You have to know what the AAA.
No, you don't have to know which of the 20 amino acids. Forget about that.
So the mRNA has different letters than the usual, A to T, C to G. It's
Yeah,
different.
the only difference is that U instead of T.
That's the only difference.
That's the only
rest
one.
is all the same.
Yeah.
Okay.
Okay? Okay, let's start this again, kind of. Okay, so here we have on the very bottom, we've got the messenger RNA that's been taken. It was made up in the nucleus. It was there ferried out into the cytoplasm. and then it was grabbed on by this RNA ribosome, okay? So the whole thing is a ribosome. And the ribosome is going to translate the mRNA into actual protein by taking amino acids and putting them in the right order. So the first thing that we should notice is that in RNA, U replaces T in DNA. So T in DNA, it's actually uracil or U in RNA. Now remember, a codon is 3 base pairs long. So each 3 base pair, in this case it's U. U for RNA replaces T for DNA. Okay, so this is the messenger RNA, and then we've got these 3 different bases. In this case, it's UUU that replaces T. Okay, and we are there, and then there's this transfer RNA. This thing here. Okay, and each transfer RNA corresponds to a certain amino acid, okay, and a certain 3-letter code. And then, of course, on the top, this transfer RNA actually is bringing in an amino acid and attaching it to a string. So the tRNAs are the interpreters of the genetic code. Now, on top is the binding site for the amino acid that it attaches to the tRNA. So the codon of the mRNA is then attaches to the anticodon on the bottom of the tRNA. And this tRNA corresponds exactly to a specific amino acid, and this is phenylalanine. So this is the universal genetic code. As I mentioned, each codon codes for an amino acid, and a codon is 3 base pairs long. In this case, we're using a messenger RNA genetic code instead of the DNA code. Okay, and like I mentioned earlier, with the ribosome, the last one that we showed was phenylalanine. So I'm going to mark that right here. Okay, that's UUU. Now it turns out that there's first bases, so UCAG, second base, UCAG, and third base. There's actually 64 possible codes with 3 bases, right? But some of them are redundant. They code for the same amino acid, and especially usually in the third base pair. So this is universal genetic code. All organisms that have ever been found on Earth use the same exact genetic code. Okay, now I'm going to also attach a video that will take you through this whole thing again, that you can watch it and it'll explain it perhaps a little bit differently. Please watch this transcription and translation video. It's a little long, but it's actually very good and should help you a lot in understanding everything. Thanks. Okay. So, how does DNA determine our traits? Well, you have the original code, right? It's been transcribed and translated into these different amino acids that make up different proteins. Okay? Now, this is the protein for, let's say, different receptors. Okay? So, the protein the receptor for tasting sour has a different protein than the protein for tasting bitter. Okay? Or the protein for tasting salty or sweet. And there's also another one, there's a taste called savory that meat has more often than not. So we can taste these different things, and those tasting is all based on what the proteins are. So this is how the protein is then, in a sense, turned into a phenotype. So I can taste salt. I can taste bitter, depending on these proteins. Some people are born without a sense of taste. Okay? It's obviously a bad mutation. So they cannot taste differences. There's also, some people can taste things that other people can't. So, let's see, what's the plant that, in Mexican food, what? Cilantro, right. Some people like cilantro. Other people, it tastes like garbage. Okay? It tastes like almost metallic. So some people can actually taste it. The other ones, they get this kind of bitter taste that's like, oh, don't eat that. Now, why that is it? Why those differences? I'm not sure. For one thing, bitter in general usually signifies some sort of toxin. So we taste bitter, and when we taste bitter, we often say, oh, that's kind of toxic. So if you were to, let's say, eat an almond, a wild-type almond, it has a lot of cyanide in it, or a cyanide compound. That tastes very, very bitter. So when you bite into an almond seed from a wild-type plant, it's going to taste bitter, and if you ate enough of it, you would get cyanide poisoning. Okay, so here's our hemoglobin gene. Okay, now there's a normal hemoglobin gene. We're going to say HbA, and then there's going to be an abnormal one for sickle cell anemia. So here's the nucleotide up on top. CTG, ACT. Right here is where the actual mutation is. Okay? So instead of having a glutamine amino acid there, for the normal hemoglobin protein, you have a valine amino acid. That valine changes the shape of that protein and makes it so it actually doesn't carry oxygen as well. But it also has another effect. It prevents the malaria plasmodium from entering the red blood cells because of the shape. Unfortunately, if you have two valines from mom and pop's DNA, that means you end up with full-blown sickle cell anemia. If you have only one copy, it means you are more immune to malaria parasites than someone with just the wild type, with glutamine. Okay? Okay. Now, genetic engineering. Genetically modified organisms. All heard about that? The difference between genetic engineering and, let's say, selective breeding is that when farmers in the past used to selective breed, they take a plant that had certain characteristics and they breed it with another plant that had similar until you get more of that characteristic. Okay? Well, when you did that, you usually selectively chose a whole bunch of other genes that kind of go along with it. So GMOs, all you're doing is you're actually taking the gene that you want and putting it into the new organism, whether it's a plant or an animal. Okay. So which cell types in your body contain the genes that encodes? Which one of these? Huh? Is it A, B, C, or D? All of them. Remember, the cell has the full genome, not just the partial. Okay, so that's a trick question. If you did this, you'd be wrong. Which cell type in your body expresses the gene? A.
What was the first question?
Okay, let's go back. So what's the name of the dimmer switch that controls or regulates gene expression promoter? So what are the two main steps that lead from the gene to a protein? And where does each step occur? So first you go from DNA to messenger RNA. And where is that? That's in the nucleus. The mRNA is then ferried out of the nucleus and into the cytoplasm, where it provides the template for the ribosome to then translate the mRNA into actual proteins using codons. A codon is 3 base pairs, and that uses transfer RNA. Okay, and if we insert a human gene into a bacterial cell, the correct protein won't be made because the genetic code is different between bacteria and humans.
Sorry, last question. The promoter region,
Yeah.
does that only happen in the area, like if it's for an eye cell, it only happens for the parts that have to do with the eye? So
Yeah.
but everything else in the DNA is saying, no, don't eat, don't eat, don't eat. So then when the mRNA is created, it's just for that.
That protein.
Okay.
Exactly, and that amount.
And that amount of apologies.
Okay. Now, in terms of GMOs, in terms of genetically modified organisms, if we take a gene, a human gene, into a bacterial cell, the correct protein that shows you why it appears that we are descended from a common ancestor with bacteria billions of years ago. Actually, sometimes do. A lot of the proteins that bacteria use are the same ones that we use. They've been conserved over time because they work. Some have been modified, so they work a little differently, but they're still the same proteins and the same genes. Okay. Before we, let's say, made insulin for diabetics, we had to get, we would have horse blood actually make the insulin. We would use horse DNA, okay, the horse insulin instead of human insulin. They both work. The only trouble is some humans actually had a allergic reaction to horse insulin. Okay. Now, what we now do is we actually take the human insulin gene. It's a little different than the horse insulin, and it's inserted into bacterial DNA like E. coli. So that exact gene, however many base pairs long, well, a couple of thousand, is taken and inserted into the bacterial genome. And then the bacterial genome will then start producing insulin, human-derived insulin. Okay? So the bacteria has become a genetically modified organism. So it's much less expensive than having a whole bunch of horses collecting the insulin from them. You end up with the right kind of insulin versus the horse insulin, which sometimes goes wrong with humans. And of course, you have happier horses because they don't have to deal with that. Okay? So, these insulin-producing bacteria are called transgenic. That means different kinds of organisms with DNA from one put into a DNA from another. Okay? So the transgenic organism in terms of insulin is the bacteria. Okay? This is the bacteria. This insulin gene is put into it, and then it's expressed and it produces batches and batches of insulin that can be then purified. Okay. The insulin protein that is isolated from the bacteria is called recombinant insulin. That means, even though the actual protein itself is indistinguishable from the human-derived insulin, it's because there was a new gene that was put into the bacteria that is now making the right kind of insulin. So that's called recombinant DNA technology. Now, because of the universality of the genetic code, we can have other organisms produce many human proteins for the treatment of different diseases. Okay? And there's a new technique out that was discovered. Bacteria, it turns out, are able to have an immune system. You might say, well, that's weird. But they do, because they get attacked by viruses. Well, that system cuts DNA, viral DNA or RNA in exactly the right place. We can now use that to precisely put a gene into another organism in exactly the right place in the genome, and not hoping it gets into the right place. Yes?
How exactly does that happen? Like, how do we put the gene inside the...
Okay, so what happens is there's actually an enzyme that actually does it. Okay, so the enzyme takes the sequence, because there's a sequence on either side that it reads, and then when it comes across that part of the genome, it immediately cuts it and then you can put the actual gene that you want to put in there, the DNA. Okay, I'm not going to go in, you don't have to know exactly how it works, but it's brand new, it's revolutionary, and I'm not going to have time to go into it, but it's revolutionary, perhaps great, but it also could lead to some really bad things as well.
So although we're getting the gene from the animal, we're converting it into a human gene, right? So we're getting the animal gene, we're putting like one of our genes and whatnot so we could use it, and then it's usable. So it's not just the whole animal gene, it's our gene.
No, so let's say we want to make a protein that we can use for, let's say, some human disease. Okay, we can put the gene for that protein, because let's say the protein that an individual has a genetic disease, they're not making the right protein. So they put the right gene in, they can then that gene makes the correct protein for the person, and then they're cured. That's what's called, what is it? DNA therapy. But it's not exactly, we're kind of getting there, and we're going to get better at it now that we have CRISPR. Okay. So here's an example. This is a protein that prevents blood clots. We have it. All kinds of animals, vertebrates have it. The gene is expressed in cells in the liver. We then release the antithrombin protein into so in other words, liver cells, the DNA in it is transferred into mRNA, then into, finally translated into antithrombin protein. Okay? Some people have a mutation It's actually a recessive mutation, so they both have defective copies for the antithrombin gene. So individuals that actually have one copy, they're still making it. Copy. Now, one thing you can do, instead of putting that right gene into the human, you can actually just put it into something that then makes a whole bunch of the antithrombin, and we can then inject that into the humans so that they're actually getting the right protein. Okay? So we actually use transgenic goats to produce a lot of this protein. We have this transgenic goat. What we do is we take a genetically modified organism, in this case it's going to be the goat, because they have been genetically altered by humans. We put a human gene into a goat. Now, where do you think is a good place to have this protein made? Where could it be collected out of a goat, most likely? Milk. So the best place to actually do it is put the gene for antithrombin into a mammary gland cell, actually a bunch of them, and then the goat will actually produce it in the milk, and then we can actually purify the antithrombin by taking the milk and separating out the actual protein. So the goat, in this case, is a transgenic or genetically modified organism, and it has a new gene for the antithrombin gene. Now, the thing is, we also need a promoter. So actually what we need to do is we actually already have a goat promoter sequence that works just as well, and it's joined together with the antithrombin coding sequence of DNA. So it's actually a hybrid gene with a goat promoter upstream and the antithrombin gene later on, and that's actually making it be expressed. And in this case, it's being expressed in the mammary glands, so it will be in the actual milk that we can then strain out or take out and purify it. It's a human gene for the antithrombin gene, but it's actually the goat promoter. In making, in this transgenic goat, what we actually do is we take the hybrid gene and inject it into a very early embryo, which will then, of course, replicate and mature into a goat, and that goat will have, that embryo will be put in a goat mother who will give birth to the transgenic offspring, and that offspring then will produce milk. If it's a female, and it will be, that will produce the antithrombin gene. So the last step is purify the antithrombin protein from goat milk. The antithrombin protein is expressed in the milk of transgenic females. The protein is isolated from the milk and used to treat the antithrombin deficient people. So they either get, it looks like they get an injection of it every so often. What the hell happened? Hold on. We've also created these transgenic organisms by inserting a gene for a protein that's fluorescent. That means it lights up, and we can actually see what cells it's been into, and we can also see actually, these fluorescent proteins, we can actually tag individual kinds of proteins with it and see where they're expressed. So transgenic organisms are used for many things, for medicine, for research, especially that fluorescent protein and things like that. Agriculture, and so with agriculture we engineer bacteria to produce growth hormone for cows, and then engineer plants to produce proteins that make them more marketable, let's say. So it turns out that bovine growth hormone, if you actually give that, inject cows with it, extra, the cows will produce more milk. So this is commonly done. But to do this, to make more of this protein, they actually use bacteria again. So we take the gene for the protein, and we implant that into the bacteria, and then the bacteria produce the BGH, and then the BGH is then used to inject into cows, and they make more milk. So in this case, do the cows, in this example, qualify as genetically modified, a transgenic organism? No. The cows just received this protein injection. There's no transfer of genetic DNA to the cow. In this example, the bacteria are actually the transgenic, a genetically modified organism. So we can give an example, but first let's go to the thing. So how do you feel about the use of RBGH, boosted milk production? Do you think that's scary? Do you think that's okay? It hasn't been shown to actually cause any problems. The actual milk doesn't necessarily have the RBGH hormone in it. Now in the past we could genetically modify foods like with artificial selection, like old-time farmers did. So if we actually look, this is a corn. The initial corn plant that was domesticated was this. It's called teosinte in the Mexican highlands, probably 10,000 years ago. And then through artificial selection, it was eventually turned into actually making big corn cobs and kernels and all that. Okay, so this is just regular artificial selection. However, one thing that we have introduced into genetically modified corn is a gene that makes the plant resistant to the most commonly used herbicide against weeds, which is Roundup. Okay, so there are genetically modified corn plants that are being planted now. You can spray the whole field with Roundup, kill all the weeds, but it will not affect the corn plants, and they'll continue to grow. Now, there's a question: is that good or bad? Is there a chance that the corn could transfer that gene to other plants around it? Because plants are notorious for transferring, especially closely related plants, one gene to another. Okay, so that could cause a problem because some weeds might actually get that gene. You'd be growing that, and then you put the herbicide on it, and it wouldn't kill those plants. So, for example, a lot of times we genetically modify crop plants. You can increase the shelf life, let's say. So there'll be some genes that actually affect how long a fruit, let's say, stays good, or how long it stays just before ripening. Or let's say you want a certain crop plant to produce a vitamin that maybe people don't get enough of, like vitamin A. So they've actually produced a golden rice that produces a lot of vitamin A, it actually produces beta carotene, which helps us make vitamin A. And that rice is being grown in places where people don't get enough vitamin A. It turns out, instead of using an embryo, we can actually modify plants with a gene gun, unlike cows injected with RBGH. Crop plants are genetically modified. In order to do this, the target gene must be inserted into the plant cell. So how do they actually do this? Well, this is a gene gun. So, introducing DNA into the plant. We have the cartridge holder, the acceleration channel, the actual genes that are then injected into the plant, into various leaves and things like that. Now, hopefully you do that in a plant that you can then take, let's say, the leaf it's been in, and you use that as a cutting to then grow a plant from that. We can also use a gene gun to put DNA into humans. I'm really not up on that. Obviously, you'd have to put a lot of DNA into humans. It seems like it would be better if you actually just did it in an embryo. Another thing that's a problem is that you've made this transgenic plant. So let's say it's you've made this transgenic corn plant that's resistant to the herbicides or to pests by using a natural, the corn plant makes a natural pesticide. All the corn that you're growing is like made with this genetically modified corn. What do you think you have in terms of this population of corn that's grown? It's actually all identical. They're clones. And so what you've done is you've got almost no diversity left in corn. If, in fact, a new disease comes along and there's no variation in terms of some corn plants might naturally have a mutation that makes them perhaps a little resistant to this new pest, then you've got this corn, rows and rows of corn that are all identical, and they'll probably be wiped out by this new, let's say, disease. So here we have kind of a showing. We've got the corn plants on the left. They're all the same. They're all have the same vulnerabilities, whether it's to drought, whether it's to flowering time, whether it's to certain pests, certain diseases. And so, like I said, if a disease came along, you'd end up with a field after field of corn plants that were destroyed, and you'd get no corn. I've attached this little video. I mentioned earlier about the new gene editing technique that's very, very precise. It's called CRISPR-9, CRISPR-Cas9. And there's this little video, you'll see what's going on. It's going to kind of transform genetic engineering, allow researchers to do all sorts of different things in a much more precise way.